Hello and welcome to SAM online class. This is Dr. Subrat Kumar Mahanti. Friends, today we shall discuss about the araOPERON as analyzed in E. coli. araOPERON is an inducible operon. It is a unique operon because the same regulatory protein can exert positive control and negative control. In araOPERON, the same regulatory protein can act as an inducer and sometimes it can also act as a repressor. So sometimes the regulatory protein induce the gene expression and other time it repress the expression of the gene. In E. coli, the metabolism of arabinoids is governed by the enzymatic products of three structural genes. These three structural genes are araB, araA and araD. These three structural genes, they encode various enzymatic products that control the metabolism of arabinoids. Figure 1 indicate about the various components of the araOPERON. First segment is araC. araC is the regulatory gene that code for the regulatory protein. And there are two operator regions, O2 and O1. These are the two operator regions. Next is inducer site. The inducer site is composed of a cap site and the I site. Next, these are the three structural genes. The transcription of these three structural genes is controlled by the same regulatory protein. That regulatory protein is encoded by the gene araC. Now we shall see when the araOPERON is induced. araOPERON is induced through positive regulation. So positive regulation of araOPERON occur when RI is bound by araC. RI is a inducer site. When only RI is bound by araC, in that case, this operon is induced. In this figure, this is the inducer site. And this inducer site is bound with these regulatory proteins. These are the regulatory proteins known as araC. When the regulatory protein bind with only RI, then the araOPERON is induced. For binding of this regulatory protein to the I site, arabinoids and cyclic AMP must be present. When the regulatory protein bind only with the I site, then the araOPERON is induced. So there is gene expression and these three structural genes are expressed that leads to the production of the enzyme that are responsible for the metabolism of arabinoids. This is about the induction of the araOPERON. So araOPERON is induced when arabinoid is present and also cyclic AMP is also present. Next we will see how the araOPERON is repressed. In the absence of arabinoids and cyclic AMP, in that case, the regulatory protein that is known as araC, this regulatory protein bind coordinately to both the O2 site and I site. This is the I site and this is the O2 site. When both arabinoids and cyclic AMP are absent, in that case, this regulatory protein araC bind coordinately to the O2 site as well as to the I site. This binding of araC regulatory protein causes a conformational change in the DNA. Due to the conformational change in the DNA, a tight loop is formed. When such type of tight loop is formed, the operon is repressed. This is the tight loop. This tight loop is formed when there is interaction between these two dimers. This is a dimer of araC, this is another dimer of araC, and you see between these two dimers there is interaction. When there is interaction between this araC dimer, in that case, a tight loop is formed and the operon is repressed, so there is no transcription. Another important point is O2 site. The O2 site is present at a distance of 200 nucleotides upstream from the I site. This is the I site. From this I site, this O2 site is present at a distance of 200 nucleotides upstream. So this regulatory protein araC, it can bind both to the O2 site and I site. For binding at either of the region, to O2 or to the I site, a dimer of araC is required. But when both the regions are bound coordinately by araC, in this case these two dimers interact, that leads to the formation of a loop. And this tight loop causes repression of the arabinose operon, because this tight loop inhibits the access of the enzyme RNA polymerase to the promoter. So friends, this is about arabinose operon. Please watch, share and like this video, and also subscribe my YouTube channel Sam Online Class, and the subscription is absolutely free of cost. Thank you very much. See you. Bye bye.